types of polyesters. One was fat and one was PET. For fat, you had an alcohol and a carboxylic acid in a way that the alcohol was named as glycerol because it was a triol, one molecule with three carbon and three OH. And then there were three different fatty acids, one, two, three. They were combining at a point where carboxylic acid or the fatty acid was giving its OH, and H was coming from the alcohol. And they were joining at a point where making the condensation between carbon of carboxylic acid and oxygen of alcohol, and three ester bonds were formed. Three ester bond. It's a limit for the fat molecule, but for polyester, which is not natural, which is a synthetic polymer, for which we named is a PET. PET does not have a limit of the ester bonds, and also it was formed by two different monomers. One was a diol and one was a dioic acid. When they joined in a repeated manner, they formed a polyester. And there were so many ester bonds you will see in one polyester of synthetic polyester. There were three, the units that we made, they were having three polyester, ester linkages. So, something that I said I will talk about it more is important here. In exams, they give you some structures like this one, or maybe a structure like this. Just focus on the point what I'm explaining here. And I told you about the kind of monomer you will also find which will have both functional group on either end. Like for PET, you have a dioic and dicarboxylic acid, two different monomers. But I'm talking about there is only one monomer, one single monomer which is repeating itself, and it is having both the functional groups on either end. So what I'm trying to tell you here is if I make two repeat unit of this, you will find that there is only two monomers are repeating and giving you two repeat units because the monomer was only one single monomer. However, here in PET, the two repeat units would be with four monomers, one of a dioic, one of a diol. This will make one repeat unit. And then for the second repeat unit, one of a dioic and one of a diol will make the second repeat unit. So two repeat units will be made out of four monomers, whereas in a molecule where you have same monomer repeated, you don't need to have four monomers. You can have two repeat units of the same monomer because this is repeated every time. So one repeat unit is here and one repeat unit is here. What is important in both of the just a minute here, you don't have to keep oxygen. The side where carboxylic acid is, you don't have to keep oxygen intact of the carboxylic side when you are making the polymer. Now, what I was focusing here is look at the direction of the uh, ester linkage. Suppose I am attaching one more unit and then I will be able to explain it better. Okay. Suppose I am attaching one more unit and this is how I have to make it properly or else my purpose will not be clear. So suppose this is your three repeat unit structure and this is one monomer containing polyester. Why we call it polyester is because we have the ester linkage here, ester linkage here. It's a polyester. This is also polyester because here we have ester linkages. You can see. repeatedly found in a polymer chain. Now, the difference between the two, you will see the sequence of the ester bond is repeated in one direction, COO, COO, and when the next unit will come, it will again be COO. Whereas if you look at PET, the sequence is flipped. Once it is COO, the other time it is OCO. This is like this. Then COO, the next time it would be OCO. The 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 pattern of the ester linkage is flipped every time whenever the next monomer is coming. This is because there are two different monomers used. So every time, uh, if first time the carbon is facing, the other time oxygen is facing, the third time carbon again is facing, the fourth time oxygen is facing. So that is why this is a very important identification to find out a natural and synthetic polymer. The synthetic polyester will have a flipped sequence of ester bond, whereas natural polyesters, and you can refer the fat molecule in this case also, the sequence of ester bond was facing only one side. It was not flipping here and there. This was not like if once you see it like this and the other time you see it like this. It's not happening here in fat. It's not happening even here in the structure which I have drawn. It is the indication that these are natural polymers. The Allah Ta'ala has kept something out of the uh, reach of human being that you cannot do. I have this power of doing. So that is why the PET is undegradable, non-degradable, because it's a synthetic polymer, whereas this natural one, whether it is fat, whether it is this one which I have drawn, they are in a particular coded sequence, and human being could not create that natural sequence, and that is why they are non-biodegradable, the one which human being have created. And the identification, if I want to find out which one is natural, which one is artificial or synthetic, this is the big identity. So the whole story that I have given you is for this, because in exam they will give you different structure. They can ask you which one is natural polyester and which one is synthetic polyester, and this is how you can find it out, and you can write it down with you because it's not there in my notes. So you can write for natural polyesters. I'm writing it somewhere over here. Let's just write here. Natural polyesters have their ester bonds repeated in same orientation. Synthetic polyester have their ester bonds repeated an alternative alternatively flipped orientation. So that's how you will find out from the structure only. Maybe they don't give you the name, the fat or the pad or any other natural name. There are only two in your syllabus which you have to learn. But by there are some actually identifications which have been explained to you, which means they understand that you will be able to know by their orientation that which one is natural and which one is artificial. That is also there in your syllabus. Hope you all have understood this point and no ambiguity about it. All right, let's move on. to the point of biodegradability and non-biodegradability, I have clarified yesterday. The definition of non-biodegradable compound has also been given because it comes for 1 or 2 marks, so you should know. And only natural polyesters will be undergoing a process of hydrolysis, which I will explain you along with the proteins. Right now, I'm focusing on the polymers, the ones which are formed by condensation. So under condensation, when we started, I had shown you this flow chart. I told you under condensation polymerization, you have fat and PET, which will be related to each other, and I told you in what way they are related. They both have an ester linkage. That's why they are related. However, one is natural and one is synthetic. Now what you are going to do, starch is out of syllabus. So what we are going to do is natural condensation polymer of another type, which has common thing called as amide linkage. I have introduced the linkage, amide linkage, yesterday. So both the protein and nylon has amide linkage, but one is natural and one is artificial, one is synthetic. So let's see how is nylon made and how is protein made. Okay, so when you talk about the nylon, which is a condensation synthetic polymer, as I told you that there is a condition for the condensation polymer, except for fat, all polymers will have this condition that they will have a carbon chain, a box of carbon atoms inside, and the two sides will be occupied by two functional groups. The two functional groups can be same or can be different. So for nylon, we are going to have two monomers. One is named as diamine. I told you the general name is diamine. You are not going to learn the whole name 1,6-diaminohexane. It's simply a diamine which has 6 carbons in between, and this is the box. And on the box, either sides are occupied by amine group. I have introduced this monomer to you in my initial class of polymer. It's a diamine monomer. It's one monomer. The second monomer you have is a carbon chain of 4 carbons, and the 2 carbons are of the functional group carboxylic acid. Hence, you call it dioic acid. The second monomer is dioic acid. And if I make it simple for you, I will be making it with a box and carboxylic acid on both sides. And if you look at the name, it says 1,6-dioic because whenever we were naming carboxylic acid, if you remember, we were including the functional group carbon in the naming. It means this is the first position and this is the sixth position, and there are 4 more carbons in between. So both, by the way, the monomers of nylon are 6 carbon containing monomers, and one is having amine on both sides and one is having carboxylic acid on both sides. So this is your simple nylon monomers, a dioic and a diamine. A dioic is one which has carboxylic acid on both end, and diamine is one which has amine group on both end. Now what happens? How do they combine? What do they make? Let's look at the formation. The carboxylic acid side, as it was giving an ester bond also, if you remember, ester bond was also using dioic. So here in the nylon also, dioic side is providing its OH, and diamine side, over there in esters it was alcohol which was giving H. Here diamines H is coming. It means diamines nitrogen will be left with one hydrogen, so NH is remaining here, and carboxylic acid will be left with a C double bond O. So if I remove the thing inside, if I just remove them, they are gone, H2O is removed. So you see what is formed, CONH. Do you see that? Do you see this link which is formed if we remove H2O from the center? So it's making CONH bond in between. Now, if I bring another unit besides this amine, if I bring another carboxylic acid with the same dioic on either sides, so its OH is gone with this H, and this side its OH will also go. So I am not making it already. I'm just making the next unit of amine which is coming. Amine was having an N and was having an H, N, and H is left because H of nitrogen and OH of carboxylic acid is gone. So this side it is also left with N and H, and this line I'll keep empty by making a bracket here. Also here I will do the same. Since this OH is gone from the carboxylic acid, what I will do, I'll make a bracket here and I'll close it to show that this is a nylon structure with two repeat units. So you can see a dicarboxylic acid box and a diamine box is making one repeat unit. Let me make it properly for you somewhere. Uh, uh, uh. Okay, let me add a slide where I can make it clear and okay, so for exam, if you consider that you had a diamine, as a monomer, and the other functional group is dioic, the other monomer is dioic. So if they are coming in a consecutive manner, alternative manner, sorry, first your diamine will come and it is going to remove H from here and OH from here, and they are going to combine this way. This is your amine box with N and H remaining in it. H is gone. From the carboxylic acid, OH is gone. So this N is going to make a bond with C double bond O. And this is carboxylic acid box. Now considering the same rule of polyesters, polyesters were left with uh, a hydroxyl group was left with oxygen. So here amine group is also left with hydrogen and nothing is there on this side because H is gone. And if you are considering considering closing this structure for polymer, you will consider that carboxylic acid has given its OH. So it is not left with its OH, it's gone. This is how you can close the structure for making one repeat unit of the polymer. This is your nylon's one repeat unit. But if you will continue it to show a two repeat unit, it means the same structure. I will copy this and I'll paste it over here. So I will start from here, this line and NH because I have to continue. So I will erase this. NH, then NH wala box, then NH of the other side, then C double bond O and its box, and then the C double bond O on the other side, and then I'm going to close it for two repeat unit. Now, till here you have the one repeat unit. This is your second repeat unit. The same repeat unit has been repeated. So this is two repeat unit structure. Remember that in nylon you will be using four monomers. Two monomers to make one repeat unit, two monomers to make one repeat unit. So here, just like PET, you have four monomers to make two repeat units. However, if you concentrate on the amide linkage. Now, if you look at amide linkage or peptide linkage, this is this. There are, you can see in two repeat units, you can see three amide linkages have been covered. And just like ester linkage, they can ask you in exam to circle the amide linkage. So you will be taking the whole NHCO thing to circle. And if they ask you to circle the repeat unit, so repeat unit would be, you can start from anywhere, by the way, but do include two monomers in that. So if I'm taking diamine here and dioic here, so I can circle this as one repeat unit. This is my one repeat unit because I'm including both the monomers in it. Both monomers should be there in your repeat unit. I hope it makes sense to all of you. Any questions? Okay. All right. So I'm moving up ahead with the protein. The protein is different in a way that its monomer is not are not two monomers. It's only one monomer, that's amino acid. However, amino acids could be different. The just like in fat you had three fatty acids, every fatty has a acid has a different R means the carbon chain. It could be 1,000, it could be 100, it could be 200, it could be 500, any carbon number could be here. So what is the difference in it? It's only one monomer, and I have introduced this monomer to you earlier. This is amino acid in a way that it has this box with carbon inside, and one side has a carboxylic acid, and the other side is an amine group. So only one monomer is repeated every time. So once only one monomer is repeated every time, you will find the sequence would always be same. So let's look at an example. This is one amino acid, this is the other amino acid. Let's suppose it's R1, it's R2. This is what makes them different from each other. So this NH2 side will be giving H from here, and this will be giving OH from here. So this N will be combined with C, and this would be your new bond, will be making NHCO. The same amide linkage you can recognize, you have just seen here in nylon, NHCO. And you will find the difference now in natural and synthetic polymer of amide, that if you bring another same unit here, same unit when you will repeat, wait, uh, this is one amino acid, CO, and now this has H and oh, C double bond O of the other amino acid will now come this side. Okay, C double bond of O of another amino acid, then another C, and then H and R, and then NH. So if I just look at the repeat units, this is your one repeat unit because one amino acid. This is your second repeat unit because second amino acid. This is your third repeat unit because third amino acid, and this is your fourth repeat unit. Over here, four monomers are making four repeat unit. Over there, four monomers were making two repeat units because they were different. Here they are same. So the repeat unit would always be one amino acid every time. And the same rule, you have to end at NH, no other H to be drawn, and C double the double bond O would be the ending. OH would not be shown. So this is going to be your protein, polyamide. And if I look at the amide linkage, 1, 2, 3 amide linkages are also here in four of your monomers repeated. There is again three linkages are there. And what's different? The difference is that the sequence of the NHCO linkage is in one direction, is in one orientation. If you go back and look at nylon, it was NHCO here. Here it was CONH. Here it was NHCO. Check it. It was nylon. It was synthetic. So synthetic is always having an alternate sequence. This is nature. Nature says no, the sequence should be same. NHCO, NHCO, NHCO. Protein is always repeating its amide linkage or a peptide linkage in one sequence. At every conjunction of NHCO, you will see that a water molecule is removed. So if they ask you to write a complete equation, you are supposed to write NH2O along with it. Is this understood to all of you? I'm going to close the chapter after only one small discussion. To Muhammad and Aima. Okay, Muhammad, you are clear? I don't find Raham. Has he left the class? Raham, are you here? Okay. All right. So, the last thing that you should know about the polymer is that natural polymers, only natural polymers, please remember it's about natural polymers, can be hydrolyzed. Hydrolyzed means break down. So, if I am saying a protein, protein was made up of what? Amino acids. So, when amino acids were joining and making protein, we were calling it polymerization. But the opposite of it, when protein breaks down to go back to amino acid, we call it hydrolysis. And this happens in the presence of water with the help of enzyme, and acid is actually providing this water. So acid and enzymes are the conditions required for hydrolysis to occur. So if I am showing this dimer of ester, if you can see, this is a dimer of ester, and I want you to hydrolyze it, break down, you can add water here. The carboxylic acid side will take H2O. The carboxylic acid side will take its OH back and will make a carboxylic acid. The hydroxyl side will take its H back and it will make an alcohol. So hydrolysis means the polymer goes back to its monomer form. So if it is happening in esters, it will be going back to carboxylic acid and alcohol. If it's happening in protein, it will be going back to amino acid. This is simple. They ask you the conditions required for hydrolysis. So you should remember acid and enzyme. This comes for 2 marks. I have seen and students were not able to answer this. So please remember acid and enzyme. You don't need to write the name of the acid. Usually it's concentrated hydrochloric acid, but you are not required in chemistry. Maybe in biology you are required, but not in chemistry. Just remember, generally you have to say an acid and enzyme is the requirement. And water is the reagent. They ask you this way, name the reagent and the conditions required. So reagent acid nahi hai, reagent is water. Reagent means something which is taking part in the reaction. So it's taking part, na, it's added over here. Water is the reagent here, and acid and enzymes are the condition. And in the presence of acid and enzyme, water is added back to the polymer and they break down into their monomers. And in polymerization, water is being removed from the small molecules and they join and make a large molecule. These two things are opposite to each other. Am I clear? Am I clear? All right. So I'm doing one of the worksheet with you. The same worksheet, as much as we could do in this remaining class time, we will be doing. And whatever will be left, I'll be sending it to you, and you can do it at home, and you can share your answers with me. I'll check and return. So we are doing condensation polymerization worksheet. You might see the repetition of the question, but it's okay. No problem. All right. You will be giving me your answers in chat box, private chat box, and not the everyone's chat. Polyesters are polymers made from dicarboxylic acids. Name the other type of organic compound used in formation of polyesters. Type of organic compounds. Yes, Muhammad, very good. Yes, Nimra, correct. No, Ahmad. Polyesters we are making. Amal, correct. Polyesters. Esters need two compounds. One is carboxylic acid. Esters, you forgot esters making. Carboxylic acid and alcohols are required to make esters.